actually published by them. But there is a lot of controversy regarding the paper they have published with Rosalind Franklin. She was a greatest crystallographer. They did recognize her, but I think she deserved more than, yeah. But still, we don't know the full story. We have two different sides of the story, but I'll not comment anything on that. But this is a controversial topic to talk about the structure of double helical DNA being published. This is published in Nature back in 1953, and yeah, of course, one of the most famous publication. This is how they have like proposed the idea of there should be a double helical structure with a minor and major grooves, and they are present in a helical form with 3.6 angstrom difference between each basis. And this was the contribution from Rosalind Franklin. Okay, and this is X-ray diffraction data from her work, which was utilized for deducing the structure of double helical DNA structure. And this is what is controversial in the whole thing. They call it paragraph 51. Okay, and if you are more interested to know about this whole story, they have a beautiful story on that. If you see in, okay, maybe before this this is the paper you can find. Okay, so if you want to know more about this story, go to Nature, which is one of the most reliable and authentic source of scientific data, and read this story. This might take 5 minutes to go through what Rosalind Franklin truly contributed to the discovery of DNA. And the first thing they started is James Watson and Francis Crick are the two of the 20th century's renowned scientists. The seminal paper from the pair of University of Cambridge, UK, detailing discovery of DNA double helix was published as a part of trio in Nature 17 years back. They were widely believed to hit the structure after stealing data from the Rosalind Franklin, a physical chemist working at King's College London. I have visited both the places, University of Cambridge and King's College London. It's a beautiful place. And they were the oldest people who told us about DNA, the structure, and even the diffraction data they observed. Rosalind Franklin had that data for a long year, but she didn't know what we can deduce out of that. So imagine if you discovered a very precious diamond, but you think it's a glass, you will not know the value of your diamond, as you know that's a piece of glass. She has that data for a long time, and then she has to share that data to someone else. Someone told people like got her the data without her knowledge or acknowledgement. But when James Watson and Crick, they got their data, they deduced the whole structure. And the saddest part is she died at the age of 37 by ovarian cancer. Okay, so imagine at such a young age she contributed to one of the most important checkpoints where we were stuck in discovering the structure of DNA as double helix. Okay, so if you're interested, you can go and read more about this whole story and about Rosalind Franklin and how she contributed. This was the original picture from this is known as paragraph 51 in science. It's a famous thing. This is X-ray diffraction from Franklin and Gosling X-rays diffraction image of bDNA. Okay, from King's College London, and they were people from University of Cambridge at UK. But yeah, it's a beautiful story. If you have time, just go through it. Now let's move ahead and focus more on the chapter, which is nucleotides and nucleic acid. Okay, and there's a point where they said discovery. I was trying to emphasize on that cost of sharing your data too early. Okay, this was the whole point of telling the story of why you should be patient to hold your data or not to publish too early. That may lead to a bigger problem solver for the entire community. People do that. People do years of research and do not publish their data. Maybe they are looking for some big stories, okay, but this is what it is, okay. So the most important part from to learn in this whole chapter is to understand the structures of nucleotides, nucleosides, and nitrogenous bases as adenine, guanine, thymine, uracil, uh, and other purines and pyrimidines. Okay, so starting with the biological function um of nucleotides and nucleosides. So there are three different terms you will have to learn. First is okay, nucleoside and nucleotide. Okay, so I generally categorize them into three parts: nitrogenous base, sugar, and phosphate. They are three different parts of a nucleotide. Okay, if you remove phosphate, it will be called as nucleoside. Okay, so in nitrogenous base, we have two types of nitrogenous base. This is also represented as N2 bases. Okay, nitrogenous bases could be purines, and the other is pyrimidines. Sugar. Last lecture we have learned about sugar. We have different types of sugar as 5 carbon pentoses, 6 carbon hexoses, 7 carbon heptoses, and so on. Okay, so the sugar we are going to see here in the structure of DNA are ribose sugar. Okay, anyone who still remember the structure of ribose? It's a 5 carbon sugar, yes, and they are pentoses. Pentoses means 5 carbon. Okay, and phosphate PO4 3 minus. Okay, so if you combine three different things, nitrogenous bases, a sugar molecule having a ribose and phosphate, you will get a structure of nucleotide. Okay, it means they are made up of nitrogenous bases plus a sugar plus a phosphate. Okay, let's understand what are purines and what are pyrimidines. Okay, so purines are two. Yes, anyone? What are the purines you have? Exactly, adenine and guanine. Okay, if you know how to remember that, that's okay. If not, I generally remember silver, which is Ag. Okay, silver, the element silver is Ag, right? Au is gold, Ag is silver, and I say silver is pure. Okay, and say purines are silver, which is adenine and guanine. And I remember pyrimidines by a famous type of reactions in chemistry, which is pyrolysis. Do you know pyrolysis? Pyrolysis is basically heating a chemical compound and will dissociate into simple singular components. Okay, it will cut down the large molecule to make smaller molecules. Pyrolysis. Pyro means heat. Okay, if you have fever, people say you have high temperature in your body. So pyro word is used for. Uh, okay, why I introduced fever is uh, a type of medicine we use called antipyretic drugs. Okay, antipyretic drugs, which means drugs we take to decrease the fever. Okay, or against, we use it against the fever, antipyretics. So pyrolysis is a type of reaction in chemistry, and I remember pyrimidines as CUT because it cuts the big molecule to make smaller molecules, where C stands for cytosine, U stands for uracil, and T stands for Thymine. Up to now, do you remember how to draw the structures of all these components? Yes, no? Do you know how to draw adenine, guanine, cytosine, uracil, and thymine? Anyone who can draw all 5 structures? And I'll buy you a drink in Starbucks. Caramel macchiato. Anyway, if not, we'll learn it today and we'll try to remember the structure um, for our, for exams or anything else. Okay, so let's learn that part first. Okay, up to here you should be knowing the basic things about it's a part of double structured DNA, double helical DNA structure. Common are nucleotides. They are ribonucleic acids because they contain ribose sugar. Okay, that's why they are called as ribonucleic acids. Okay, denaturation and annealing, we'll learn these steps later on when we already know the structures of those molecules. Chemistry of nucleotides and mutagenesis. Mutagenesis is the process of generation of mutation. Okay, the process of generating mutants are called mutagenesis, and the chemicals or component which generates the mutations are called mutagens. Okay, anyway, functions of nucleotides are nucleic acids are they are energy molecules, which is ATP, adenosine triphosphate, NAD, which is a component as cofactor for different enzymes, and they are used in signal transduction as cyclic AMP. This CAMP is cyclic AMP. AMP stands for adenosine monophosphate. Okay, ADP stands for adenosine diphosphate, and ATP means, what is ACP means? No, this could not be a thing. So I should say CTP, cytosine triphosphate. Okay, or GTP, guanosine triphosphate. Okay, so there could be several variations of those different permutation combination of all the nucleic acids with different nitrogenous bases. But they are known for storage of genetic material as DNA. They are also in the form of mRNA, which is messenger RNA. How many types of RNA we have? How many types of RNA we have in the molecular biology? 3. I got to answer 3. And they are, can you speak up? Exactly. It's mRNA, tRNA, and rRNA. Ribosomal RNA, transfer RNA, and messenger RNA. Okay. So this mRNA is also contains nucleic acids, and we'll see how they are transcribed. I think all of it are written over here. Okay. Anyway, we'll learn about the structures of purines and pyrimidines. Okay. Few thumb rules to understand the structure of any purines or pyrimidines. You have to learn how they are named. Okay, and it's very easy once you know how to name them. Okay, so the first thing to remember is the order of the numbers we assign to each atom. Okay, so the first rule is always start with nitrogen. Always start with nitrogen. Okay, if you have purines, always go anti-clockwise. Okay, in purine we always numbers anti-clockwise. Okay, so I said first thing is start with nitrogen. The second thing I said is this numbers as anti-clockwise. Okay, so see, I started here 1, so this is anti-clockwise, right? And if I represent this is clockwise. Okay, if you don't know, you see the clock, it's running this way. So when you draw structures like this, you can rotate your hands for sure. This is clockwise for me, but anti-clockwise for you. You get that? So this is clockwise for me, but anti-clockwise for you. This is anti-clockwise for me, but clockwise for you. It's all about the imagination, how you observe things. But But whenever you draw the structure of purines, they always go anti-clockwise. Okay, this one has the same rule. You have to start with nitrogen, but they go clockwise. You see that this is clockwise and this is anti-clockwise. Okay, but yeah, if you have to observe, if you have a second ring, it will always go clockwise. See that 7, 8, 9. Okay, I'll draw this two more time to make sure you understand those structures correctly. Okay, you have to understand how basically they are named and how to remember them, and then you'll be able to draw the structures of all nitrogenous bases. Okay, so I'll explain these things about later on once we learn the structures. So I'll start with adenine. Okay, so always remember adenine and guanine are purines. Okay, and I said they are named anti-clockwise, starting with nitrogen, right? So I'll start from here. Let's change the color to red. So this is 1, this is 2, this is 3, 4, 5, 6, 7, 8, 9. Okay, first part is clear. When you have purine, you will start with nitrogen always. Okay, and you have to go clockwise or anti-clockwise? Anti-clockwise. Okay, and the second part will be clockwise for sure, 7, 8, 9. Okay, so you know this thing, you can remember, and i'll I'll explain which atoms will be at what position. Okay, so I always remember this NCN, CCC, NCN, NCN, CCC, and NCN. Okay, can you repeat after me? It's NCN, CCC, NCN. Okay, so it's NCN and NCN, and in between you have 3C, which is 3 carbon. Okay, can you repeat after me one more time? NCN, CCC, and NCN again. Okay, so let's see if we have done it correctly from the number 1, which is NCN, CCC, NCN. Is it correct? NCN, CCC, NCN. Okay, so this rule is always there: NCN, CCC, and NCN. Okay, I repeat this so many times that you remember the structures. Okay, it's NCN, CCC, NCN. Okay, so I remember adenine as 6 amino purine. Okay, please write down these names because it's very important to remember the structures. I always remember adenine as 6 amino purine. Okay, why? At 6th position we have a amino group. Okay, and that's why we remember adenine as 6 amino purine. Can you confidently say that you can draw the structure of adenine by your own? The first rule is always start with the N, go anti-clockwise with NCN, CCC, and NCN. Okay, let's understand the double bonds arrangement. Okay, so you will have alternate double bonds 1, 2, 3. Okay, and then alternate double bonds with one nitrogen and carbon, and that's it. Okay, can you draw this structure one more time with me on your paper or whatever you have? Okay, let's draw with fresh, nothing, white page. So I'll start with nothing. I'll draw N, C, N, C, 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 and N, C, N. Cool. Let's connect all the atoms, which is 1, 2, 3, 4, 5, 6 membered ring, and then 1, 2, 3, 4, 5 member ring. Okay, so I always say it has a 6 member ring and one 5 member ring. Okay, and it is 2 ring structure. Okay, and let's understand the position of let's number them one by one. I said always start with nitrogen. I said 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, I have to add a double bond. Mostly double bonds comes with carbon and nitrogen. One double bond, two double bond, and three double bond. Okay, always remember, when you have a common atom, you have to write a double bond. Okay, this is common between those two rings, and you have a double bond over here. You have to leave a single bond, then double bond. You have to leave a single bond and double bond, and this is how you remember. Okay, whenever you have a bridging atoms, you write a double bond, and alternate to that is a double bond, alternate to that is a double bond. This is called conjugation in chemistry. Okay, let's understand where should be the other double bond. Leave one single bond here, the other bond double bond would be here. And I say, if you have a purine, you always say this is not purine but this is 6, this is not adenine but 6 amino purine, and 6 amino purine is nothing but adenine. Can you remember the structure now? Yes, I need some confidence in your answer. Can you draw the structure of adenine now? Yes. Let's understand the second structure, which is guanine. Simple, very simple, sir. You have to remember the same thing I said. If you are drawing a structure of purine, you have to remember, start with nitrogen and go anti-clockwise. So the first thing is, start with nitrogen from here. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. So after that, how to remember the guanine is, I say 2 amino 6 oxy purine. 2 amino 6 oxy purine is guanine. Okay, you have to remember this name, and you will remember the structure for sure. I can guarantee you that. If you remember 2 amino 6 oxy purine, you remember structure of guanine. Okay, so why did I name this? Because we have the second position amine group and sixth position oxy group. Okay, so if you don't want to remember this way, always remember you already gave amine position 6th for one purine, and now it will be on second position. Okay, you just have to switch the position of amine group 2 from 6 to 2, and then you have oxy group at 6th position. Okay, so can you repeat this after me? Guanine is 2 amino 6 oxy purine. Okay, one more time. Guanine is 2 amino 6 oxy purine. Okay, so can you help me draw the structure of guanine from the structure of adenine from here? I didn't draw it. This is amine 6 amino purine, which is adenine. Can I draw the structure of guanine here? What you have to do is you have to erase this one, and you have to add oxy group. So once I add the oxy group, there's a problem. What's the problem? Carbon is having 2 to 4 and 5 bonds, which is not possible. Okay, so what we have to do, we have to erase this double bond. Okay, and I said 2 amino, right? So this is now 2 amino 6 oxy purine. Okay, 2 amino 6 oxy purine, and this is not adenine but guanine. Cool? Any confusion? No? So one more time, adenine. I want to know the scientific or IUPAC name. Adenine is 6 amino purine, and guanine is 2 amino 6 oxy purine. Okay, and you can remember the structure of, or you can draw the structure of both adenine and guanine simultaneously. One more time, can you see the structure? It's start with nitrogen, NCN, CCC, NCN. Okay, Second, NCN, CCC, NCN. Okay, and this is 6-oxy 2-aminopurine, and this is 6-aminopurine. That's it. Can you see there's no double bond over here? Why? Because carbon cannot form 5 bonds altogether. Okay, that's it. There's no other difference between adenine and guanine. Okay, let's move ahead, and I think we are able to understand how to draw the structures of all purines, which is adenine and guanine. Let's move to the second part, which is drawing the structures of pyrimidines. Okay, I said pyrimidines means pyrolysis. You have to remember the three names, which is cytosine, uracil, and thymine. Okay, so these names are simple names. You don't have, you will not get idea of the structure they are forming, right? The names I said you have to remember so that you can know the structures of those names, right? The first is cytosine. I said when you draw the structure of pyrimidines, you have to remember start with nitrogen and clockwise. Okay, still the same rule, NCN, CCC. Easy, right? There's no other thing. NCN and CCC. So it means it's a single ring structure, right? The purines are double ring structure, and this is simple ring structure. Simple. Can I move back and see how they are numbered? See this? This is numbered clockwise. Okay, NCN and CCC. You can also start from here and say NCN, CCC. No problem. Okay, so I'll start from here naming 1, 2, 3, 4, 5, 6. Okay, and I will say N, C, N, C, C, C. Okay, can you repeat after me? It's N, C, N and C, C, C. Okay, always start with nitrogen and go clockwise. Okay, so this is how we are going clockwise. Similarly here too, 1, 2, 3, N, C, N, C, 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 clockwise. One more time, N, C, N, C, 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 clockwise, right? So we figured out how to name and number the atoms in pyrimidines. Okay, let's understand the naming of those pyrimidines. So can you guess the names of those pyrimidines by now? Just amine and oxy group. It's easy. 2-oxy pyrimidines. 2-oxy 4-amino pyrimidines. But we'll do a slight change. You are correct. But we'll say 4-amino 2-oxy 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 pyrimidines. Why? Any guess? Because this is O and this is A, and according to IUPAC, alphabetically ordered, we have to write the names, right? That's why I wrote 4-amino 2-oxy pyrimidines. Okay, can you remember that? Okay, now we'll learn uracil, and we are skipping thymine for a second. Okay, we will learn the second structure, which is uracil. Okay, anyone who can tell me what the IUPAC name of uracil could be? No, for uracil. We stopped for thymine for a bit for a reason. Okay, 2,4 2-oxy, right? And 2 we use di 2,4 dioxy pyrimidines. Okay, 2,4 dioxy pyrimidines are uracil. 2,4 dioxy pyrimidines are uracil. Write it down. 2,4 dioxy pyrimidines are uracil. Just one more structure to learn, and then You are good to go with nitrogenous bases. I remember thymine as methylated uracil. Just write it down. Methylated uracil is thymine because this is how nature created them. They have done a methylation in uracil and made a methylated uracil as thymine. Okay. So I always call thymine as methylated uracil. Hello, who is there? So methylated uracil is thymine, correct? Can you name thymine in IUPAC? It's very simple and similar to uracil, right? And if you haven't written yet, uracil is 2,4 dioxypyrimidines. Okay. So anyone who can name thymine similarly? 2,4 dioxy 5 methyl pyrimidines. Correct. You are getting it. 2,4 dioxy 5 methyl pyrimidines. Okay. Or can I name it other in a different way? In comparison to the uracil? Any guess? What's the difference from the uracil they have? Is a methyl group at 5th position. Can I say thymine is 5 methyl uracil? Yes or no? Simple, right? 5 methyl uracil is thymine. Okay, you just have to look at the structure and try to remember it. Okay, so 5 methyl uracil is thymine, right? Can we draw one more time? White page theory. Easy to remember, right? Should I wait? You noted everything? Okay. Let's try to remember the structure starting from scratch. First, NCNCCC. Okay. I'll start with here NCNCCC. Okay. 1, 2, 3, 4, 5, 6. Okay. One more thing, position of double and single bond. Okay. It's easy. You don't have to remember everything. You just have to be cautious of following the rules of chemistry. Okay, and that's it. Can you see it could be a double bond here? It could be a double bond here. So how to understand which is incorrect? Just do the incorrect thing. I always do that. If you want to know whether this thing could happen or not, just do the incorrect thing and you will definitely understand this cannot happen. Okay, I'll put a double bond here and you will see it's not correct because carbon is again having a 5 bond. So I'll erase that. Okay, so I'll erase that, and now you have a double bond in a similar sense than other way. Two double bond here, and there's no double bond because of the oxy group. Okay, similarly there cannot be a double bond here. See that there cannot be a double bond here, not possible, not here, not possible. So there is only one double bond in structure of thymine. Similarly in uracil I cannot put a double bond here or here. See that? Any doubts? No. So the only structure having two double bond is cytosine. Okay, because it has only one oxy group. If you have two oxy group, there cannot be two double bond. Two double bond can be removed from the structure. Okay. So yeah, this is the position of nitrogen and carbon. I'll name them 1, 2, 3, NCN and CCC. Okay, I'll put double bond everywhere for now because I don't know whether I'm gonna do for cytosine, uracil, or thymine. Okay, so can I draw the structure of cytosine from here? You have to remember the scientific name, which is anyone? 4 amino 2 oxy pyrimidines. Okay, and I'll draw the structure NCN and CCC. Okay, 1 2 3 4 5 6. I said fourth position is amine group, which is NH2, and second position is oxy group, one double bond here, one double bond here. Okay, clear? Correct. Second structure NCN CCC. Okay, uracil, I'll draw it first because I know thymine is methylated uracil. Okay, I said uracil is, one more time, scientific name 2,4-dioxypyrimidines. So 1 2 3 4 5 6. Okay, 2 position oxy and 4 position oxy, 2,4-dioxypyrimidines. One more time, NCC. Cool. 1 2 3 4 5 6. I said it's similar to uracil, so I'll do 2,4 dioxy and 5 methyl, right? Fifth position is methyl, which is CH3. Okay, pyrimidines, or 5 methyl uracil. Cool. So I believe you can draw structures of adenine, guanine, cytosine, uracil, and thymine by now. Okay, one more try. One more time, I'll try to see whether you still remember the structure of adenine. Right? Can you tell me the scientific name of adenine? 6 amino purine. And guanine, 2 amino 6 oxy purine. Cytosine, it's right here. 4 amino 2 oxy pyrimidines. And uracil, uracil is the easiest one, 2,4-dioxy pyrimidines. And thymine is 5 methylated uracil, 5 methyl 2,4-dioxy pyrimidines. And thymine is 4 amino 2 oxy, no, not thymine, cytosine. Cytosine is 4 amino 2 oxy pyrimidines. Okay, clear? So I think you are able to remember the 5 different structures of purines and pyrimidines up to now. Let's move ahead and see about the nomenclature. Most important. There's only one problem here, and that's it. Nomenclature of I said you have nucleoside, you have nucleotide, you have nucleic acid. Okay, and what's the difference between all of them? Nucleotide is sugar plus base plus phosphate. Okay, ribonucleic acid. Okay, sorry, this one is a polymer chain of nucleotide, nucleic acid. Okay, nucleotide is sugar plus base plus phosphate. Nucleoside is sugar plus base. Okay, your base is basically nitrogenous base, and this nitrogenous base could be purines or pyrimidines, and your purines are A and G's, and pyrimidines are cytosines, uracils, and thymines. Okay, so let's understand the adenine first. Adenine could be adenine plus adenine plus your sugar could be adenosine. Okay, you get that? This is adenine, the structure we have learned, and you have a sugar which is ribose sugar. Okay, so adenine plus ribose sugar is adenosine. Okay, you add a phosphate and then it will be adenylate, which is sugar plus base plus phosphate. Did you get that? If not, let's see this structure, okay, of a nucleotide. Okay, and you will learn it better. This is a nitrogenous base. Anyone who can tell me what is this nitrogenous base? Adenine, thymine, cytosine, guanine, uracil. If you are not getting this structure now, I might be a bad teacher. Adenine, thank you so much for keeping my words. Okay, this is 6 amino purine. Can you see that? Sixth position is amine group. Yes, sir. 1, 2, 3, NCN, NCN, CCC, 7, 8, 9. Okay, and sixth position is amine group. So this is definitely adenine. Okay, so this is purine and this is adenine. Okay, which is a nitrogenous base. This is a phosphate. This is phosphate, and this one sugar. Do you still remember this carbohydrate class? Yes, this is first carbon, second carbon, third carbon, fourth carbon, fifth carbon. So this is a pentose, yes or no? Because it has 5 carbons, right? And this is a ribose. If you don't believe me, go to the carbohydrate class and see the structure of ribose over there. Okay? So there is two terms. One is ribose and the other is deoxyribose. Okay? So this is pretty obvious from the name. Deoxy means they definitely have removed the oxygen from somewhere. Okay? And by somewhere I mean mostly second position or third position. They will definitely write it down. So can you see there is no oxygen in the second place? So can I call it 2-deoxyribose? Yes, because at second position you remove the oxygen, and we can say this as 2-deoxyribose sugar. Okay, and that's why they are named so. Deoxyadenylate, which is a nucleotide. There's nucleoside and nucleotide, and the only difference is phosphate group. Okay, so if there is no phosphate group or I remove phosphate group from here, this will be deoxyadenosine. Cool. This will be deoxyguanosine. Let's understand this way. If you have a guanine and you add a ribose sugar, this is guanosine, right? If you add a phosphate again, it will be guanylate. Okay, and imagine if you have a second position oxy missing, which is deoxyadenosine, deoxyguanosine, deoxycytosine. No, because cytosine is already there. Can you see that there's a difference? So if you have adenine, you can write adenosine. If you have guanine, you can write guanosine. But this is already sine. You see that? Can you see a difference of cytosine? They are named very differently. If it could be something else, we can definitely name them as cytosine. Are you okay? Who is this? A ghost? Okay, welcome to the biochemistry class. Let's learn nucleotide, nucleosides, and nucleic acids. So cytosine is already a base, and cytidine is a nucleoside. Okay, so I can put this question anywhere. Which one of them is not a nucleoside? Okay, and I'll put somewhere cytosine, and the name is very obvious. It looked definitely as a nucleoside, but it's just a nitrogenous base. Okay, and that's it. And when I started this slide, I said there's a one problem over here, and that this is a problem. Not problem, but this is something different, right? And I said thymine. Thymine is thymidine, and uracil is uridine. Okay. Similarly, cytidylate, deoxycytidylate, uridylate, thymidylate, or deoxythymidylate. Their deoxy means second position oxy is missing. Okay, a nucleoside, if you have adenine, it could be DNA, RNA. If you have a ribose sugar, it's RNA. If you have a deoxyribose sugar, it's DNA. So when I say DNA, which is deoxyribonucleic acid, means second oxy is missing. Okay, that's why they are DNA. And the other one, which is RNA, has all the oxygens in the ribose sugar. That's why they are called RNA, which is ribose nucleic acid or ribonucleotide. Okay, so DNA and RNA are named. Why? Because these are the components they have, oxy and deoxyribose sugar. Okay, you understand how DNA and RNA are named so after this class? Yes or no? Yes, sir. He's getting it right. Once you get it, you will feel it. If not today, the other day, as Dr. Narayan say. Okay, so I think we are here. We're up to here. Good. Let's practice some more structures based on that. Is this purine or pyrimidine? If I don't get an answer, I leave the class. Purine or pyrimidine? Pyrimidines. Why? Because this is a single ring structure, right? Single structure are pyrimidines. And what is this? Anyone can tell me? I'll hide these things. I cannot hide it because this is 2,4 dioxy 5 methyl pyrimidine, or I would say methylated uracil is thymine. So this is thymine. Thank God I am getting answers now. This one 4 amino 2 oxy. I'm not supposed to talk. You are the one who should be doing all this. The first one is cytosine. Okay, this one I'll get 3 seconds. 1, 2, 3. This is 4 oxy 2 amino. Yes, guanine. This is adenine and this is guanine and this one is thymine and this is cytosine. Okay, so I think you are at least capable of recognizing any purines or pyrimidines up to now. And this is deoxyribose sugar. This is also deoxyribose sugar, deoxyribose. So all of them are deoxyribose, deoxythymidine, deoxycytidine, deoxyguanosine, and deoxyadenosine. Okay, I'll see one more time. Are they deoxy? Yes or no? No. So that's why they are not deoxy but RNA. All of them are ribonucleotides, and these are deoxyribonucleotides. So basically, these are DNAs. Okay, and these are RNAs. So can you understand the difference between DNA and RNA now? Yes or no? Yes. So let's see, this is a nucleotide which is adenylate. Okay, guanylate, but uridylate. Why? This is uridylate. Why? Because this was uracil before, and this was cytidylate because this was cytosine before. Okay, do you remember these structures now? 6 amino purine, yes sir. 2 amino 4 oxypurine, yes sir. Guanine. This one is 2,4 dioxy 4 amino 2 oxy cytosine. Thank you. So they all have phosphate. Can you see that? And that's why they are nucleotides. Okay, let's move on and see. Up to that point was all you have to understand about the structures of DNA, RNA, adenine, guanine, thymine, uracil, and so on. Okay, now comes the part which is more conceptual. Those were for the understanding. If you understand and practice one more time, just one more time at home, and I can guarantee you will remember the structures for whole your life. Just go home, sit at home, and give me 30 minutes and practice all those structures today itself, and you will remember those names for life. So it says polynucleotides. Poly means polysaccharides. Poly means many sugars. Polynucleotides means many nucleotides. And if you already know what are the nucleotides, it contains a nitrogenous base plus a ribose sugar and a phosphate. Okay, it's called phosphodiester linkage. So imagine if you have a nucleotide here and a nucleotide here, the bond is known as phosphodiester. I always look for the name. It says phospho and it says diester. Di means 2, ester means a type of functional group, right? 2 ester group, which is OCOR. Okay, and then you have a phosphate. So imagine you have a phosphate from one nucleotide and a phosphate from other nucleotide. So they remove a water molecule in between, and they call them phosphodiester bond. Okay, I'll show you how they make phosphodiester bond when they attach nucleotides. So I will compare again how carbohydrates, how proteins, and how nucleic acids, which is three major components in biological system, are polymerized. Okay, I'll start with amino acids in protein. Do you remember the bond when two amino acids are combined by removal of a water molecule known as condensation? No way. I talked about proteins. Proteins, two amino acids combining one removal of water molecule. Amino acid 1 to amino acid 2. Bond is? Come on, I need that answer. You can do that. It's a covalent bond, but name is different. Oh, no. Two amino acids are attached with peptide bond. Peptide bond, peptide bond, peptide bond. Okay. And two sugars are attached by glycosidic bond. Imagine 1 2 glycosidic bond, 1 4 glycosidic bond, 1 6 glycosidic bond, 1 1 alpha beta, 1 alpha 2 beta, 1 alpha 6 beta. Come on, I taught them just a few days ago, right? And the third is nucleic acids are attached with phosphodiester bond. One is peptide bond for proteins, glycosidic bond for carbohydrates, and phosphodiester bond for nucleic acids. Okay, get that? So let's see how a phosphodiester bond is formed. It's easy and it's obvious. Uh, okay, so phosphodiester linkage or phosphodiester bond. This is just a basic information, as DNA have more stability than RNA. And what are the reasons? Anyone? Anyone? Basic difference between DNA, RNA. It has a deoxy. So this